Good morning, everybody. So I'm Itsik Pe'er from Columbia University, and I would like to first thank Zar and Stacy and everybody for getting us back in person.
If you go to the mic on the desk, point it in the other direction. Okay, that didn't work.
Maybe not. Just a minute. Here. No, I turned off the wrong one. But okay. This does not work, but this does.
Does it work?
Can people? So. Does this work? Does this work?
Yeah, but
Okay.
much feedback.
Much feedback?
Much feedback, yeah.
So you know what? Here, I'm gonna. Here, just let me turn on the volume.
Try again?
Yeah, try.
This is muted. The lavalier is muted.
Okay, try that.
Can people hear?
Yeah.
Can people hear me at the back? Back. Wonderful. Thank you. All right. Even more reason to thank Zar and everybody. It's great to be back at CGSI, and I want to tell you a bit about our work of last couple of years, looking at the microbial dynamics. And at this stage, I hope people here know that most of your cells are not yours, and definitely not. Most of your genes are not yours. They belong to bacteria that we farm in every nook and cranny in our body. And one effective way to collect data from such microbiomes is to collect samples from subjects and then sequence a universal gene, most commonly the 16S ribosomal RNA, that comes in different flavors for different species. So when you look at the reads, you can just count them, and you know how many reads of which bacterium you have in each of the samples you've collected. Now, unlike my I guess previous field of activity, which is inherited genetics, the microbial content of your microbiome can change. So it actually is very meaningful to take multiple samples from a particular proband. This is a particular sample from a paper a decade ago where they had been following transplant recipients before and after the day of transplantation, and whenever they collected samples and sequenced the microbiome, they were able to get the composition that of the gut community. And one thing that you can observe is that for this particular individual, a few days after transplantation, there's a particular strain that really took over the entire community, which is kind of the reason why we would like to follow these individuals in order to monitor, in this case, the appearance of vancomycin-resistant enterococcus and try to predict this kind of event. This also highlights that the data that we get here is from reads that sample from the microbiome, so we only get fractions as the information that we typically obtain, unless you go through quantitative PCR, which you can, but it makes things less streamlined and cost ineffective. So, this perhaps gives us some sense of the different complications and challenges that rise when you're looking at time series microbial data. Obviously, the data by definition is temporal. It's also noisy because it's biological data. And we're looking at compositions rather than absolute data points. The data is often sparser than we would like it to be. And if I have time, we'll also address the fact that while we often treat the gut, for instance, as a site for the microbiome, at the microbial scale, the gut is huge, and there's a lot of geography going on along and across the body niches that we often abstract as single points. So there is a spatial component as well. So let's start dealing with this. Well, I'll go through several pieces of work developed in the lab over the last few years, maybe not delving too deep into each and every one of them, but the references are available to you, so they're all actually published. So the first issue to address has to do with the noise of the bacterial sampling and processes. And when you think about that, what we're trying to go after are the biological processes that govern the dynamics of bacteria. And these temporal processes tend to be continuous and smooth. So the ground truth process, the different compositions along the time axis, need to behave like something like this. In this particular case, there was some initial conditions that are somewhat aberrant or perturbed, and there is in this particular simulation there was some convergence towards a steady state. But this is a far cry from what we actually observe in practice. What we actually observe are read counts that, taken at face value, would lead us to estimate percentages of operation taxonomic units that are much noisier versions of this ground truth. So our goal would be to take these data and go through each of the taxa and sort of figure out what we think is the more appropriate curve that describes the actual trajectory, so we get a better approximation of the ground truth. Now, this is a very generic smoothing lines, smoothing data points is a very generic data science task. People have been doing that with lowest curves or whatever for ages. But when we looked at the data, we realized that we can do better if we have models that actually describe what we think the data, what we think are the rules that govern the data. So what's the probabilistic model that we have under the hood? Well, we first assume that the actual biology is some kind of a Gaussian process. So there is a vector at each time point that describes the actual ground truth state of the bacterial community. So how much from each strain or species we have in our sample, and the vector at the next time point is normally distributed around the previous state with variance with a covariance matrix that's scaled by the length of time that had elapsed between these time points. So this is what we think. So this is how we abstract away just the absolute state of the community. And for reasons that maybe may become clear later, we believe the right way to think about that is that the additive process, additive Gaussian process here, is not directly is not directly controlling the frequencies of different bacteria, but rather the logs of these abundances. So the actual frequencies are more related to these states exponentiated, and then with an additional Gaussian noise on top of them. We don't observe these directly. What we so we actually observe reads that are sampled from these frequencies. But when we're trying to develop this model, these kind of models have been suggested in the past. One thing that we found missing is that there needs to be some way to deal with taxa that are present or absent in the system at all. So on top of this quantitative part of the model, we added a dichotomous vector of the species that allows species to appear, say if there is contamination or infection, and allow species to disappear if they get eliminated, because then they're sort of out of this game of frequency once they're eliminated from the mixture. So given these binary vectors that can change along the time axis, we can now draw reads from the species that are present at a particular time point at particular frequencies, and get an overall distribution, multinomial distribution of the actual observed reads based on these probabilities that are normalized probabilities given the quantities and the presence or absence of particular taxa. Now, so this is the model, and if you remember Nick's presentation about Nick Mancuso's presentation about variational inference, this is kind of a model that lends itself to variational inference because you can approximate this distribution in a very decomposable manner, and which is what we're actually doing, and that allows us to effectively infer the parameters of the model, which is what we're after. If you don't remember Nick's presentation, there are videos. There's a video tutorial in CGSI from 2019 where David Knowles gave a tutorial about variational inference, and if you don't like that, there's another video where I gave a tutorial about variational inference. So you have no excuse. Beyond that, I'm not going to go into the details of the method. So let's just convince you guys that it's actually working. So this is R squared between a ground truth data and the inferred trajectories. Higher and closer to 1 is better. Blue is kind of a straw man model that doesn't have any connection between the different time points, just a Dirichlet prior multinomial. Red and green are Mallard and TGP Coda that are other methods that are trying to offer similar kind of models for this task, and we're doing significantly better across most of the time of the simulation models. So noise simulation models added onto two distinct data sets. One that describes infection by C. difficile, a common hospital infection, and another that models change in diet.
What is the vertical axis?
The vertical axis is R squared, so correlation to between the estimated trajectory and the actual ground truth. Okay? So we're doing somewhat better, but I think more importantly, because of that variational inference that you all now remember or will go watch the videos, we are able to do that effectively. So the other methods have been trying to conduct inference using MCMC type methods. You have to deal with integration in order to infer the parameters, unless you approximate the posterior and do variational inference, which allows you to be effective and fast, even when you have lots of data points and lots of taxa, where other methods really break down. The y-axis is runtime in minutes per single sample. If you are trying to work on data sets that span, say, more than a month of daily sampling, including hundreds of samples or more, this y-axis can become a real nuisance if you're not fast. Yes?
So what's the typical number of taxa that you might see in samples? Like, is 50 the right number, or is it higher or lower?
What's the typical number of taxa that you have in a sample? So it depends how low in frequency you So you consider the reads. You can go down in so you typically have thousands of reads per sample. So in principle, you can have thousands of taxa, but you probably don't care about things that are very, very rare. There are results that really point to, like, so there was a cell paper a couple of years ago highlighting an obesity important bacterium that's at a sort of 1% range. So you probably want to sample down or to consider all the bacteria, at least down to that range. Even though if you look at some of the papers, people are sometimes abstracting this away and saying, okay, we want to only look at the top 10 or top 20. Part of it is because of computation constraints. Hopefully we can remove this consideration and actually look at the dynamics all the way down. More questions. This is a good opportunity for questions.
Can you maybe just briefly, now that people understand what it's doing, kind of go over the method?
Okay.
I know it's a complicated method, but maybe just the key intuition.
So the key intuition for variational inference is that you have so that the distribution of the data is often very complicated. So you need to infer lots of parameters that are connected to one another in a convoluted way. But variational inference builds on that distribution being approximable by a fake distribution that is more amenable to computing efficiently. Specifically, this approximate distribution replaces, in this case, these PT variables with fake PTs. So PT tilde is a common notation, and that allows the posterior distribution to be decomposable. When it's decomposable, you can infer parameters of the of this approximate distribution effectively because they don't affect one another. So that's kind of the an example, if you will, of the general principle of variational inference.
And you're saying that the difficulty in this whole problem is because every point is tied together to every other point in the time point, right? Because you're saying that there's like a small change over time, right?
Yes, yes. So for this recording, yes, the difficulty is that all the parameters are connected to one another. So all the data points are connected to one another, and the parameters affect one another. So the entire inference process is a complicated distribution. We want to simplify it, and that allows us to do that.
So in your simplification, you actually disentangle or detach the parameters from one another? I mean, so you optimize each one separately?
So we optimize so it's like
Hill climbing.
it's like more it's like an EM algorithm, right? So people are often more comfortable with EM. What does EM say? Well, we're not going to optimize the distribution, the parameters of distribution. We're going to optimize the value, the expected value of the parameters given where we are right now along the optimization trajectory. Formally, what we're doing is optimizing the expected log likelihood of a fake distribution of the data, which is the same thing that variational inference is also doing. Okay?
Tiny question.
Yep.
You said the fake distribution. Any assessment about the relationship between the fake distribution and the real distribution?
So like in EM, improvement of the log likelihood of the conditional log likelihood in the fake distribution is guaranteed to improve your log likelihood for the actual distribution. So we rely on that and iteratively improve.
Yeah. Okay.
Okay?
Yeah.
Thanks. Yeah, this is important. Yeah.
You have an underlying ecological model here, right? This is the X naught, XT, so on and so forth?
Can you say that
You
again?
have an underlying ecological model that's describing like the true community dynamics, correct?
Do I have an underlying ecological model? So for this part, not really. So this is kind of ignoring any particular ecological ecological model. We're going to introduce an ecological model right now. Is that so? Should we do that? All right. So, ecological model is coming alongside dealing with the compositional aspect of the data, the fact that even if you have the data in D dimensions here, D is 3 different phyla, the actual data that we register is compositional, so it's 1 dimension fewer, because we only have fractions of the reads from each of the phyla. So we need to have that in mind, and why this is important is because a common ecological model to think about dynamics of complex systems of multiple species is the Lotka-Volterra differential equations. People may be familiar with them from predator-prey kind of systems. You have rabbits and foxes. If you have too few rabbits, then the foxes starve, and so on. And so the same kind of principles you can envision as governing the dynamics of bacterial systems. So the rate of change in the at particular time t of the abundance of that particular of a particular taxon I, well, that depends, or that multiplies that particular abundance of taxon I at time t by, well, first of all, by the growth rate of that particular taxon, and then by positive or negative interactions that this taxon I has with any other taxon J at time t. So while bacteria do not usually directly prey on one another, they do compete, so would have negative interaction. The more of you that there are, if I'm competing with you, I would have a harder time growing. But they also have cooperative relationships. So if you are excreting metabolites that I would like to consume, then the more of you there is, the better I would be able to fare, the better I would be able to grow. So there is some kind of a matrix, Aij, that defines these interactions between each taxon J and each other taxon I. In actual systems that we're interested in, we're not only interested in the natural ecological effects and the interactions between bacteria as they grow without disturbances, because we want to take advantage of the malleability of microbial communities and perturb them, interfere, hopefully for the better, or model perturbations for the worse. So there are particular perturbations that affect the particular perturbations that we can apply at particular time points, and they would have different effects. Each perturbation P may have a different effect on each species I. So this is the kind of equation that makes sense in absolute abundances. Unfortunately, we typically don't have these absolute abundances, so we need to change these distributions, these equations, and create a compositional version thereof. So if we replace these absolute abundances by fractions, we can we have one dimension fewer. Suppose we choose one particular species to normalize against. Well, there are other ways to do that, but that may be simplest to talk about. We get a new equation that's very similar. So instead of growth rates, we have relative growth rates and relative effects of intervention and so on. There is one annoying factor here, which is the overall total community size at each time t, which is unfortunately not a constant. What we're going to do is try to ask how bad is it if we approximate that as a constant, and to what extent can we still rely on these modified equations, the compositional equations, and then solve them with an assumed fixed overall composition. This is despite the fact that this is not really constant for the data sets that I mentioned earlier, the diet data set, the overall composition varies about an order of magnitude, and that the nice thing about this is that even given that, we're still able to get estimated coefficients that are very good approximations of the actual coefficients that you would get from the absolute data. In this particular data set, we had absolute data because they actually did bother to run qPCR and get absolute abundances. So we were actually able to have ground truth. So the coefficients for the interaction matrix and for the perturbation matrix, in this case a perturbation vector, because the perturbation is a change in diet, were actually very good. The growth rate is fine, even if not great. For the other data set I mentioned, again, the overall total composition varies about an order of magnitude. The results are somewhat less impressive. Still, the effect of the actual perturbation, which arguably is the major point of the experiment, is relatively accurately inferred. Yes.
Are these matrices of interactions A and perturbations B fixed unknown, or are they changing over time or unspecified?
Are these matrices?
Are they like constant over time, like things that you're trying to estimate but you're assuming constant?
So are the matrices constant? Yes. So what changes in time is the potential introduction of a perturbation. Under this model, what can change in the dynamics of your rabbits and foxes is that all of a sudden the forest burns down or something. But the actual interaction is presumably actual biology that is fixed. Questions?
So when you say this is less impressive, what is less impressive?
What is less impressive? The correlation, so the inference of the parameters here, is correlated, but with like R squared of.
This is R squared quite high.
This is
This is quite high.
well, depending on your standards.
Yeah,
If your standards are 0.993, then yeah. But we do have we do infer. Thanks for the kind words. Decent correlation between what we infer in the composition model and what could have been available with the qPCR data, so with the actual full abundances. The whole point of this is really introducing the compositional equations and asking to what extent can we still do that in a compositional environment. Okay. So this is kind of inference of parameters. One way to interpret whether the inferred parameters are impressive, not impressive, good, great, is the extent to which they allow us to infer ahead from initial conditions, and they do. So here, lower is better. So the y-axis is root mean squared error in the prediction for the next time points, and these compositional equations, compositional logical theory equations, are doing better than trying to fake them in some way by different transformations to the compositional vectors that have been suggested in the literature of just trying to change to normalize the raw data rather than to have equations on the raw data that are taking into account the dimensionality that's one dimension fewer, and all the relationships are compositional. These are different. So there are different normalizations that have been suggested in the literature. They're doing better than the straw man of doing nothing, but still having the right model. And this on real data, we're able to do better. And so that's for the diet data. This is for, and these are for two other two other data sets where qPCR data had been available, so we're able to have these comparisons. So I'm gonna use a couple of minutes just to quickly tell you about the other projects without going into detail, addressing the sparsity and the geography of microbial communities. So because we're only able to sample particular time points and are often interested in a lower resolution of the dynamics along the time axis, an effective or an important piece of the toolbox had been the reliance on metagenomics, so getting shotgun reads from microbial genomes. And this was proposed by Talkorem and colleagues a few years back, observing that when a particular bacterium is growing and dividing, DNA is replicating, so you have more reads near the origin, more DNA, therefore more reads near the origin of replication than near replication termini. If you align these reads to the genome, you should see a peak near the origin and a trough near the replication terminus. The bacterial genome is circular, so you should so the end here connects to the beginning. And this leans on a theory that dates 50 years back that ties the replication timing and the generation timing to what we think, what we project this peak to trough ratio between amounts of DNA should be. But the theory was developed under kind of constant conditions. We generalized it a bit, but more importantly, we're able to develop again a variational inference method to estimate these growth parameters conveniently for cases where we actually when we actually don't have a reference to align the reads to. This still happens, but you just don't know where the reads are coming from. We're able to still do that by looking at kind of reads assembled on the fly. We don't know the genome, we don't know where they lie, but we can ask, well, what if there is growth here? So there should be peak to trough. So, because if there isn't, we don't care. If there is, there should be there should be a slope between replication origins and replication termini. So if we order the different bins of sequence according to their overall abundance in multiple samples, we should be able to get an approximate ordering and therefore an approximate rate. We're able to demonstrate that it's doing well in practice. The new method is able to accurately to much more accurately estimate peak to trough compared to the previously developed and published optimization by Tal. This all leans on Tal Korem having moved to Columbia and having become a close collaborators of ours. So we're able to do that with his lab. Let me skip the example across one data set because I want to just mention the geography of gut microbiota. This leans on work from Harry Swang's lab at Columbia, who was able to develop this nice assay, MapSeq, that essentially shreds the mouse gut into small bits. So you can have not one gut microbiome, but with one barcoded collection of reads, you get different contents of the microbiome from each of these pieces of the gut. Now the not so nice thing is that you lose the ordering. You don't know where these puzzle pieces are coming from, so you have to resort to computation. We have modeled that as a Gaussian process along the presumed axis along the gut, and explored whether this is the kind of model that's relevant, or whether we should be looking at additional dimensions rather than just the length of the gut, governing the real data, the actual observed data. It depends on the real data and is a noisy version thereof, and we're able to order the different pieces here along the presumed length of the ileum, where the data is consistent with a single axis. But for the distal column, we're seeing significant support to having separate axis, so one axis along the gut, but also one that's likely a radial axis between the gut that's closer to the to the axis of the gut and different cavities along the gut. Yes.
So you do have some information about, okay, they are like anchors, right? Where you know this was taken.
Do we have anchors? No, we don't have anchors. The labels here are post hoc, figuring, okay, this corresponds to what's been seen in the literature. That's this consistent with the lumen, this consistent with the mucus, and so on.
By the signature, by the expression, by what you see in the actual data.
Yes. This is relying on the labels are reliant on the actual data. Yeah.
But the other axis, you have no confirmation.
The other axis, we don't have a confirmation. Okay. So a lot of that. So the primary person that had been driving this work in the lab is Tyler Joseph, now at Regeneron. Other people involved are Philip Chlensky, currently in the lab, and a brilliant then undergrad, Amipo Sarkar, who's now starting his PhD at Princeton. Lots of thanks to the microbiome labs at Columbia. Specifically, data here was from Tal Quirm's lab and Harris Wang's lab. And thank you all for your attention.